Welcome to the Ethics of Paediatrics Genomics seminar. I'm coming to you from today from the studios at the Royal Children's Hospital. And I'd just like to start by acknowledging the traditional owners of the land where we're meeting from today, and that is the Wurundjeri people of the Kulin Nation. And I would like to pay my respects to their elders past, present, and emerging. So thank you for joining us today for this symposium. My name is Chris Skinjel. I've got the honour of introducing the seminar today. I'm a team leader in the Biomedical Ethics Group at the Murdoch Children's Research Institute, and I'm also an investigator on the grant, the Centre for the Ethics of Paediatric Genomics, which is why we're bringing you this symposium today to highlight some of the issues we've been researching. I also want to start to thank today the Children's Bioethics Centre, in particular Lynn Gillam and John Massey for co-sponsoring this seminar for us today, and also just acknowledge that we are funded by an MRFF, a Medical Research Future Fund grant, so we're thankful for that support as well today. So just give a bit of background for what the type of things we're going to be talking about today and what we're sort of interested in is to start from the realization that genomics is starting to have a huge impact in medicine and in medical care. And we can see genomics applications really starting to have a big impact across 4 domains: research, screening, diagnosis, and treatment. So in research, we've been able to genomics has been allow us to sort of find some mechanisms that are important in a lot of diseases, which can then help us sort of find treatments. With screening programs, we've been finding that genomics is increasingly being used to be able to identify healthy people that are at particularly high risk for developing certain diseases. And this is sort of an exciting time in this area. I note that in Melbourne is going to be hosting one of the first large sort of screening programs looking at healthy adults and using genomic sequencing to look for their risk of cancer. That's a study that's been run out of Monash. So again, this is a different area where genomics in medicine is starting to really play an increasingly important part. For people who are sick, genomics is having an increasing impact in diagnosis. So for helping find the reason why people are feeling sick. And this is particularly being particularly revolutionary for sufferers of rare diseases who have often been sort of left behind in medical progress, and sometimes will go on journeys for years and years without being able to find an accurate diagnosis for their for their disease. And genomics is really revolutionizing diagnosis in this area. And finally, we've seen that genomics is having a big impact on treatment. So people respond differently to different drugs, to different treatment interventions. And what genomics allow us to do is to sort of find people who might respond slightly differently to a treatment, and then to be able to tailor treatments towards those individuals. So this is sometimes called pharmacogenomics, and I've was on the FDA website just yesterday, and there are now sort of dozens and dozens of recognized pharmacogenomic interactions. So we see how genomics can also sort of influence treatment and has this really broad use in a variety of different areas of medicine. But with this great progress have arisen a number of ethical challenges and complexities. Most of note probably is consent. So giving informed consent to a genomic intervention or to a genomic test. Informed consent is, a real cornerstone of medicine, of appropriate medical practice, but it really requires people to be able to understand the potential implications of having a test, to be able to understand the potential repercussions of what they might face as a result of the test. But with genomics, it's increasingly hard to go over what the potential outcomes of a particular test are. And there are other complications too. In fact, that genomic information, when you test someone, you're not just testing them, you're also revealing information about their family members and about others. So this makes it very hard for people to be able to sort of give informed consent. And it also makes it very hard for clinicians to be able to give the patients enough sort of information, enough time that they can sort of make an informed, reflective decision about whether to have a genomic test or not. And this really touches on another sort of interesting ethical concern, which stems from how far do we look into the genome? When you do genomic sequencing, when you're trying to find the result of a test, there's lots of other information in there that might not be of direct relevance to the reason the person was feeling sick. So there might be information about their risk for other diseases into the future. And there's this really interesting question about, how far do we look once we're doing a genomic test? How far do we look for these other attributes that are in the genome? And this touches on a related concern, which is that we might be sort of over medicalizing people through genomics. So for people who are not sick, who are healthy, they might not have a complaint about their body or about their sort of state of health, but genomics might be able to find them because all our genomes are different and there are lots of different variants in there. So there's this real concern about this overdiagnosis and medicalization of perfectly healthy people. And overriding this general concern is the general question of what do we do with this data that we're generating? So genomic data has potential use in research, potential use for individuals through their life. And how we store and manage this data, therefore, has really deep implications for the health system in general. And it's really important that we have these mechanisms for using this data efficiently and effectively. So this is the sort of state with genomics, medicine, and ethics generally. But what happens when you add children to this, when we're talking about pediatric genomics and ethics? And a general rule of life, I think, is that when you add children, things always get more complicated and slightly more challenging. And in pediatric genomics ethics, that is no different. There are these great sort of ethical tensions that just exist in pediatrics about sort of the barriers or sort of the limits of parental consent. How much should parents be able to decide for their children? When does the state, when do clinicians need to sort of intervene for parents or intervene against parents? And this raises very, very interesting sort of ethical challenges when you apply it to genomics as well. So some of the specific sort of challenges that we find when we look at pediatric genomics ethics include how do we adapt these ethical frameworks for the unique contexts that arise in pediatrics? So one of the best examples of this is when we're looking at critically ill children and the rapid genomic testing, which is technology that's been really pioneered here at the hospital. And it's a remarkable sort of feat that they're doing. But it raises these interesting questions because it's use in genomics in this completely new sort of domain where the children might be very, very sick. It might be a time-sensitive nature to the genomic test. And that's very, very different to how we've been doing genomic tests differently. So it really raises these sort of deeper questions about how do we adapt our frameworks, how do we adapt our practices in genomics for these unique clinical contexts that are arising in pediatrics. Related, a question that arises is how far do we peer into a child's genome? So if we're looking at the genome of a sick child, do we just look for the genes that are directly related to the illness that can explain their illness? Do we look for other disease-related genes, or do we just look for anything? Because we know that a child's genome can reveal not just information about their illnesses and about their health, but against their sort of different traits, maybe their personality traits. So it's this really question about how far do we peer in and, what do we look for while we're in there? And a related sort of question is, what sort of role does genomic sequencing have in newborn screening? So as we said, we've been talking about doing genomic sequencing for very, very sick children. Does it have a role in healthy children as well as a sort of screen? And these sort of are the three sort of challenges that we're going to be sort of looking at today, and that hopefully we're going to be generating some more discussion about these interesting areas. So I'll just go quickly through our program for today. So in a few minutes, we'll be starting our first session, which is looking at the topic of rapid genomic sequencing in critically ill children. I will break for some morning tea. We're then going to have a session chaired by Dania Viers on additional findings in pediatrics. And Then we'll have lunch, and our final panel discussion, which I'm really looking forward to, is the question of, are we ready for genomic newborn screening? And Lynn Gillam will be chairing us, and we've got a great panel, which you can see there, and it's going to be a very, very exciting discussion. So I am going to quickly now go through to our first session, which is rapid genomic sequencing in critically ill children. We have 4 speakers for you today. I'm just going to go through their bios quickly. So speaking first will be Zonitza Stark. She works for Australian Genomics and the Victorian Clinical Genetic Services in Melbourne. She completed her medical studies at the University of Oxford in the UK before training in paediatrics at the Royal Children's Hospital in Melbourne and in clinical genetics at VCGS. She completed a Master's of Bioethics at Monash University in 2013 and was awarded a doctorate in clinical genomics from the University of Oxford in 2017. Zonitza is passionate about the application of genomic technologies as a first-tier test for the diagnosis of rare diseases in children and has authored over 150 publications and leads several national genomics projects, including a rapid genomic testing program for critically ill infants and children. Following Zonitza, we will hear from Katie Arkell. She is a Senior Project Officer with Melbourne Genomics Health Alliance, implementing genomics into clinical practice across Victoria. Katie has a Bachelor of Science, a graduate diploma of genetic counselling, and recently completed her Master of Genomics and Health from the University of Melbourne, in which her research project explored ethical implications of ultra-rapid genomic sequencing in the paediatric setting. Following this, we're going to have a case discussion with Kirsten Boggs, who is a professionally trained and certified senior genetic counsellor who has worked as part of the NHMRC-funded Australian Genomics Initiative since 2016 and has contributed to a number of research programs and publications. More recently, Kirsten was involved in the recruitment, consent process, and genetic counselling of hundreds of paediatric rare disease patients across the Sydney Children's Hospital network, with a recent focus on the MRFF-funded acute care project. And finally, we have Julian Copeland, who is a lecturer in bioethics at Monash University and holds honorary positions at the University of Melbourne and the Murdoch Children's Research Institute. His research interests include human tissue ethics and the ethical implications of new technologies.